Good morning. We are going to start. Well, we are going to start with this session number 5, late breaking news and poster highlights. We have 8 talks, 8 speakers, and the first speaker is José Luis Cantero from Universidad Pablo Olavide, and he's going to talk about decreased total antioxidant capacity is associated with lower glucose consumption in temporal lobe of normal elderly subjects. Please, Luis.
Good morning, everybody. First of all, I would like to thank the scientific committee for selecting our work for presentation in this session, so thank you very much. Aging is a very complex phenomenon under the influence of genes and environment at a ratio of 30-70. That means that factors as nutrition and lifestyles may account for about 70% of different trajectories of aging. In this context, it would be important to have global, modifiable, and minimally invasive biomarkers able to detect different levels of cerebral vulnerability during the aging process. Many studies have identified the accumulation of oxidative stress in brain tissues and also in the bloodstream as one of the most common mechanisms of aging. However, there is no data supporting the link between global antioxidant status and aging-related changes in cognition and brain integrity. In order to fill this gap, we have measured total antioxidant capacity by using the ORAC technique, cognitive function covering memory, attention, and language, cortical thickness using high-resolution 3 Teslas cerebral MRI, and cortical glucose consumption with FDG PET scans. This was performed in 100 normal elderly subjects. With this study, we wanted to test the hypothesis that decreased total antioxidant capacity predict poor cognitive functioning and also lower integrity of the cortical mantle in the elderly. We first found that decreased total antioxidant capacity levels were significantly associated with bilateral reductions of different regions of cortical glucose consumption in different regions of the temporal lobe, extending from the inferior temporal cortex to dorsal regions of that lobe. In the right hemisphere, this relationship between antioxidant status and glucose consumption was extended to extrastriate cortical regions. We didn't find any relationship between total antioxidant capacity measured in blood and cortical thickness, suggesting that this relationship was mainly affecting synaptic function rather than structural integrity of the cortical mantle. We further found a significant relationship between total antioxidant capacity and higher levels of homocysteine. And this result was obtained after removing the effect of age in the statistical model, suggesting that this relationship between antioxidant status, measured in peripheral blood, and homocysteine was mainly due to cerebrovascular factors, as for example, small vessel disease due to age, rather than age itself. And finally, we didn't find any relationship between total antioxidant capacity and cognitive function in the elderly. However, when we split our subjects in those who had memory and non-memory, those who were memory and non-memory complainers, we found that subjects with memory complaints had significantly lower levels of total antioxidant capacity when compared with non-memory complainers. So in conclusion, Our study revealed that lower levels of total antioxidant capacity are associated with the presence of suggestive memory complaints, higher levels of homocysteine, and bilateral reductions of glucose metabolism in the temporal lobe of elderly subjects. And then our results support interventions based on healthy lifestyle aimed to enhance the antioxidant capacity in late life. So that's all. Thank you very much.
Hi, nice talk. I have one question. Is there any, based on the literature, that this total antioxidant capacity correlates with any measure in the central nervous system? So that can infer that this is also what's happening in the brain. And the second is, do you have any measure of inflammation, peripheral inflammation, that can also explain this, correlates with the measure?
No, the answer is no to the two questions. We don't have any correlation or any analysis supporting the relationship between these levels, these lower levels of total antioxidant capacity, and other measure of change of some parameter in the central nervous system. And we don't have yet inflammation parameters that we can assume that could be related with these levels of peripheral total antioxidant capacity. So.
Very nice results, José Luis. What is your interpretation or explanation for these particular areas showing the reduction in the PET signal? It's quite unusual to see anatomically so extrastriate areas associated with this, particularly in the context of memory or subjective memory complaints.
Yeah. And in a previous study, we showed that it was a strong relationship, very robust, between changes at a temporal lobe level and suggestive memory complaints. So perhaps this is a kind of mediator effect between this total antioxidant capacity, suggestive memory complaints, and this lower integrity of the temporal lobe. It's something that we should take a look.